the third session. I don't think I need to ask this question, what is sequence design? I know I've been doing that before, but I think at this point we've kind of figured this out. So the main idea of sequence design is, given a protein backbone, we want to predict a structure that is likely to fold back into that backbone. So we have a structure, maybe say it looks like this. What sequence can be used to refold it into that shape? That's really all we're asking. So why does this work? Well, one of the things that I like to point to whenever we're curious about these protein structure sequence structure relationships are that, like, the protein structures are super degenerate. And by what I mean by that is that there's a lot of patterns in structure space that are recapitulated by many different sequence patterns. In fact, this study in 2016 showed that around 600 terms, which are just like tertiary local structural motifs, will cover around 50% of the structural space at a sub-angstrom level. It goes logarithmically past that, but the fact that 600 terms can still cover the 50% of the database is quite remarkable. And I think it really helps point to the fact that nature is really just kind of reproducing some things that it's seen over and over again. And it's kind of just reusing these bits and pieces to rebuild more new complex machinery, and you can mix and match them to kind of build something more unique as well. We also know that proteins that share homologous structures, they can share homologous structures even at a very low sequence identity. A common threshold is around 30%, and proteins with even 30% can still be called homologs, which is still pretty remarkable. So this really all points to the same fact that the structure really can lend itself to multiple different sequences, and we can try to figure out those sequences by learning these structural patterns. So it's not going to be like deterministic. We're not going to have a single sequence that best fits this structure, but multiple sequences should be able to ideally adopt the same structure. So as we've done before, I want to talk about some design metrics that are associated with sequence design. The first of which is sequence identity, which is pretty self-explanatory, but it's just the percentage of the positions that have the same predicted amino acid as the native amino acid. It's a percentage, and it ranges from 0 to 100, and it is length dependent, of course. Another one that you're probably less familiar with is called perplexity. This is a measure of how confused the model is when it's trying to make its selection. So one thing you could kind of, that's a little bit more intuitive to grasp is essentially this question: how many choices is it choosing from when it's trying to make its decision? So if it's looking at a residue position and its perplexity is like 6, it's trying to choose between 6 residue types to place there. So it's a measure of like how confident the model is in that backbone structure and how it like corresponds to sequence space. So if there's like perplexity of 1, it's really confident that only one amino acid should fit that structure really well. So it's
like a mathematical formula for that?
There is. So you take the negative log likelihood and you exponentiate it. So it's a positive value. It's not length dependent, and there's no real units there.
So isn't there kind of a limit in the sense that in a lot of positions you can have multiple amino acids that are valid?
Yes.
Like, do you want like a 0 or whatever the best, like where every position has a simple choice, or is that like almost bad?
I don't know. What do you think? What do you guys think?
When we were talking about perplexity, I just keep going back to the thought of like sequence conservation, and some products are like variable across the sequence diversity. So I was going to ask, has anyone looked at perplexity versus like a family sequence conservation across structure? Because there are some spots where they're pretty variable, and maybe not super important to the function of how this family evolves. So my take on Jonah's question is like, I don't really want like a one single, this is the one. I'd rather have something that can give me a diverse sequence.
Yeah. So I'm not sure if people have looked at that. And part of the complexity there is that this perplexity is model dependent. So like you'd have protein MPNN's model prediction, models perplexity for the specific residue in the specific protein. So it's a little maybe difficult to look at, but if you're restricting your investigation to like a specific protein fold, specific protein family, it's maybe a little easier to look at, but I'm not aware of any studies that do that. But yeah, anybody else have a take on Jonah's question?
Well, is it correlated with, like, if you were generating a structure with AlphaFold, whether those two, the backbone and the structure you generate, are like, are those ever correlated, or is it like correlated to a point and then just has to?
That's a very interesting question. We'll see a little bit of that in ligand MPNN, but there is some correlation there. Yeah.
I think that a higher possibility might be good if you're like construct like a network of possible ancestors for your protein, so you can like make a better tree.
Yeah, yeah. If you have that diversity, it is more easy to create trees and also be able to sample the same structure really, but maybe different sequences. So yeah, my answer to that would be, I don't think we want something that is ultra confident that this is the right answer. Because we know that protein structures are so degenerate, that we know that that cannot be right. So there are some problems with that. It's similar to the idea of like AlphaFold predicting a single conformation. We know that's not right, because proteins are dynamic, they're very flexible, but it's still kind of useful. I don't know, yeah.
Is perplexity on a residue position or on the entire sequence?
You can measure it on both. I think most people average like the average perplexity over this test set or something like that.
Sometimes the perplexity also has to do with how well trained the model is on a particular residue. So if it just doesn't know very much about the residue and try to stick it very close, it'll give you
Yeah, because you obviously don't want your perplexity too high, because if it's just making random choices, it has no idea. It hasn't really learned anything about the sequence structure relationship. But if you have too precise of a or perplexity value, then maybe the models just overfit to your training data. So there is kind of like a nice balance you want. I don't know what that number should be, and I would argue that it's up to debate and may be backbone dependent and family dependent.
So, say in like a de novo design campaign, you're trying to develop like a binder to a target. Can you use perplexity as a heuristic? Because the way it sounds, it's like I could possibly look at it on a per residue basis and determine if I'm actually producing enough sequences for a specific region of that protein. Like if it's giving high perplexity, would that necessarily say that I've done enough sampling in that region?
So if there is a high perplexity for a specific region, I think that could point to the fact that maybe the model thinks that many different sequences can adopt that same specific conformation. It could also reflect that the model just has no idea. It has never seen this before, so it's just making a random guess at what the answer should be. So I think it's difficult to exactly say what the meaning behind the perplexity is in that case, kind of like what's the cause of this model's reasoning. So I would maybe caution a little against that just because it's a little difficult, yeah.
So I can follow up, and how is it used currently in typical design?
Yeah, so typically lower perplexity is typically better just because right now I don't think we're getting to the point where these models are overfitting to the data. You can easily overfit them to the data, but when we're averaging it over the entire test set, we typically aren't really worried about if you've trained your model well enough, it shouldn't really be a concern about the overfitting, because like we'll have the data, the information about like what the training set and everything was, so we'll kind of know if it's overfitting. But yeah, typically people just kind of look for the lower value. But as we've discussed, that might not be the best case once we start getting models that are maybe a little bit more accurate. But I think there will always be kind of a balanced region of perplexity that we're interested in.
I would say one thing on the region versus residue is that a lot of the sequence design models are autoaggressive, so they depend on how confused you are about the surrounding context. So it might really know what's going on at one residue, but if like the perplexity adds up around it, then it might just overall.
Okay, and the last one I wanted to talk about was the designability. We've really already seen this before in the backbone generation step, but I just wanted to bring it back around because it really kind of combines the backbone generation and the sequence design. Once you have a backbone, you want to create a sequence for it, you throw it through a structure prediction network, and then compare how well does that match the backbone that you were designing that sequence for. And once again, you kind of just measure it as a percentage. Yeah. So why would you want to do sequence design? I don't think we really need this part, but I'm going to go through this quickly. You can do full partial design, so redesign the entire sequence. You can fix part of a protein and redesign the rest of it. You could also do biased design, so you may have a preference for maybe a positive charge in this specific region, or maybe you want this core to be very hydrophobic, so you can kind of bias the model towards these predictions, but it might not necessarily agree with that. So it kind of allows the model to make its own decisions too, but you kind of put a prior distribution that pushes it towards a certain direction. You can also do multi-state design. So you may have multiple conformations for your protein that share the same sequence, but exist in multiple conformations, maybe at different rates or something like that. So you could bias the sequence predictor to take into account both states at the same time. So it'll look at the fold of one state and the fold of the other state and kind of just average its predictions in order to choose something that will favor both. And very similar to multi-state design, you can also do symmetric design. So now you have multiple copies of the same protein and say we want these two residues to be, well, if we have multiple copies of the same protein, we want the entire proteins to be symmetric. So you tie every position to the corresponding position in the other copy of that. But you can also do it at not necessarily a whole protein level. You can tie individual positions together if you want that. But that's part of symmetric design. Yeah.
Is there a capability to, say, given a protein that partially embeds in a membrane, to design the dissolved exposed portion and the membrane exposed portion with different weights?
Yes. So they've trained protein NPNN on a few different ways. One of which is this residue level membrane token. So you can essentially tell protein NPNN that part of the structure is in the membrane. So treat it like it's in the membrane, whereas the rest of it would be maybe solvent exposed. Any other questions? Yeah.
Could you explain more about the multi-state design? Like, what does multi-state mean?
So multi-state is kind of multiple conformations of that same protein. And so it doesn't always have to be positive design either. So you could say, so if you have like a fold switching protein, it adopts multiple conformations. You would want a sequence that is favorable for both conformations. So that's kind of the multi-state design, is positive multi-state design. You want something that is favorable for both of these states, but you can also do negative design. You can say, hey, I really don't want this state, and so bias away from it, whereas promoting this state. So there are multiple different ways you can do that. You can combine different constraints like that. So it can get really complex, but that's kind of the main idea.